Hi, good afternoon. Welcome to our testing. Please let me know if you can hear me, please. Just doing some live testing. I'm going to be doing some tissue portal today. All right, great. I'm going to, I'm in the middle of sterilizing some plants, so bear with me. Excellent. I hope that it's not too loud. I have the hood is on, so hopefully it's not too noisy. Okay. Okay, let's see. Let's see some questions. So, can I freeze the plant growth regulators after reception? What do you mean? Well, yes, you can freeze the plant growth regulator. So whenever you have your stock solutions, um, you can freeze them, but you just keep them in the fridge. That's good enough. Um, how much will ppm cover up for cleaning of the plant material? I mean, you want to PPM is like an insurance, but I mean, if you don't sterilize the plants, I mean, you still see the. Um, yes, I know I'm live. I just doing a test streaming and I'm working on some of this stuff. But yeah, I know I'm live. I just a little quiet right now. But yeah, no, when you're working on with a PP with for sterilizing plants, you want to have a proper sterilization, and PPM will help you um, prevent the growth of fungus and bacteria. But you still need to sterilize your explant using a bleach. You see all the test tubes that I have here? All of these that I'm going to be doing today. I have a 63 test tubes. I have my Eiger right here. So if you are interested in getting some test tubes, we have all of these test tubes available on the website. And anything that I'm using here today, all the Agar, the media, all the test tubes, Everything is available on the website. Okay, you need to check your sterilization protocol. What plants are you working with? Flying ice? No noise at all? Is it no noise of the food or is no noise or like you cannot hear me? Like 1 moment. Okay. All right, let's get it started and then use some actual tissue for control. Yeah, you having a lot of um, problems with moisture on any plant. It doesn't have to be only with cannabis. I use filter paper for my containers, controversials or micropores. Yeah, you can like make a hole on your flask or like you see here, like we have these test tubes. The test tubes have a filter on it, so these are great for preventing excess moisture on the inside of the tube. Oh, okay, I see now you cut off, so the audio is okay, right? Yeah, well, you know, ya, bueno, ahorita nada más una prueba. Sí. Oh, okay, okay. Yeah. Too much water here. I like to keep a little bit of sterile water on the tray, so I have so many eggplants, so they don't dry out. All right, it has been a while since I actually done tissue culture. So let's get going. And if you are not aware, I know you saw my previous videos, but I used to move full time to Washington DC, so expect to see me a lot more often. So maybe live streams here, Instagram, or a lot more videos. So any experience? Okay, so like if I was using some, well, actually I don't know what plan is that. I'll have to do some research on that plan because I have no idea. Any explanation relation of the biocopters? Yeah, so we have for the biocouplers, we have an automatic method, which is a prototype at the moment. So soon we want to have a release, um, an automatic process to flip the biocouplers. So, so that's coming pretty soon. Right, let's work these plans out pretty quickly. So you see, as I cut the trim of the plant where the bleach has sterilized the plant or the tissue to death, I just trim the end and then I just place it and I just set it up where, well, actually I'm pressing on buttons. I need to place them the way around. That way, because I'm right-handed, they are on the right way for me to place in the media. Process pretty quick. I can do close to 50 to 100 test tips in an hour. But yeah, you want us to do more like live streaming, please leave us a like on it so we know like you are enjoying or take some value of doing like this live streaming. They are a lot very easy for me to do because I can use go live pretty much any time. I'm now working full time in the lab, so I can just pretty much come over here and then just hit the stream and then go live. And I'm not checking the chat at the moment. I will come back to your questions here after I finish streaming these plans. I hope you have a good some good holidays. We are my start of the year has been very eventful. Now with the move, full-time move over now to Washington DC, I'm very excited. We are all very excited that we're going to be working here full time. So you are more interested in learning more about tissue culture. This weekend we have a tissue culture masterclass. It's online, so you can join us for anywhere in the world. to be online this weekend. So make sure to check planceltechnology.com and then you can sign up for the masterclasses. And then you can use my code FP10. You can get a 10% discount. Not only for the class, but also for anything that is on the store. We also have another class coming up in February. That one is with Bill Graham. That is on mid-February. Also, you want to check that one out as well. Yeah, I know from like my last video, the title was like I'm leaving. So actually, I know people they thought I was actually leaving, but probably didn't saw the entire video where I was leaving from the university. I used to work at the university, and now I work full time plan sales. So you will see me here more often now. You can see how fast this process is. As soon as I'm done trimming the edge where the bleach has killed the tissue, I will start moving into all of these test tubes. I have 63 test tubes to do. And if I have any extra explants, I'll grab some larger containers that I have. And I'm sorry I'm not looking at the comments right now. I just want to make sure I finish in all of these plants before I do that. All right, I'm almost there. Almost there. So how's everybody doing today? Like we just decide to do like use a stream, use a testing as a test stream, see how it goes. People want me to go live more often, we can do that. So I'm done streaming. All right, let's see. Let's take a look. Um, thank you. Interesting bottle caps. Do you insert the filter paper yourself or the pre-made? Oh, like this. They come with the manufacturer. So our manufacturer, we already come out like this. So this is how it comes out. Sí, las clases son en persona solamente son en Washington. Posiblemente en un futuro vayamos a algunos otros lados, pero en este momento solo son en Washington. How long does it take to see something grow? Well, depends on the plant. Well, you say from leaf. So also, I mean, depends on the plant, but from leaves. A lot of the times you grow from leaves, then you get callus first, and it will take some time. I mean, you have to be patient. At the beginning, it will probably take, I don't know, maybe about a month to you start seeing some callus grow, and then like a 2 3 months where you can start seeing some new shoots coming out. So yeah, it can take a little bit of time, but uh, can you tell us more about introducing callus? What plant? Tell me what plant. Look at my water. All right, let's take time. I'm about to start doing, like I say, 63 test tubes that I have here. Let's see how long does it take. Uh, 2:30 p.m. Viendo el stream, uh, looking at the stream, it's probably going to take me longer than I would do it that normally would take. That's okay. On nice shape. Cannabis and peppers. What do I dissolve my plant growth regulators in sodium hydroxide instead of potassium hydroxide? I'm not sure. I mean, I you can do both, but it's sodium hydroxide is the what we use the most. Do I do mushroom culture? No, I do. I personally don't. That's Shay. Shay, that's he's the one who does the mushroom cultures. I want to learn. I need to. Now that I'm here, can have time for him to teach me how to do mushrooms. That's his stuff. What species I'm working with? Yes, with that one. You know what I'm talking about. Give you a little close up there. That's the plant I'm working with. And this agar feels really good. It feels really good. I really like these test tubes. This is actually the first time working with these test tubes, uh, and I really like it. Again, you can find this on the website plantcelltechnology.com store. You can use code FP10 for a 10% discount. I mean, you see how fast I'm going, which I'm actually taking it quite easy. Use a steady pace. Will I do more education for embryo rescue? You know what? A lot of people have asked us about the embryo rescue like lately, like in the last month or so. I don't know. Is that you that who's been asking us? Because it has been quite popular lately. So so yeah, sure. I mean, I can do some videos if you want to. How many grams per liter is the egg milk? This is 8 grams per liter. 8 grams per liter, and it makes a really nice. Feels really good. Like it's not too soft, and it's definitely not too hard. It feels pretty good. This is our supreme grade agar again for plant cell technology. We have old seeds. I'm sorry, what I got lost. What kind of seeds are those? You want to sprout those old seeds? We have a couple of videos on how to sprout old seeds using hydrogen peroxide. So you may want to check those out if you haven't already. We need a video on storing genetics in tissue culture. I mean, this is what I'm doing right now. I mean, you can see I'm doing tissue culture on test tubes. Um, you can see one of these racks here. I show you when I finish this rack. Well, you see that it doesn't take that much space. I can do is I can take this rack, place it in the fridge, and give it maybe put a light on it, the fridge, and they will store here for a while. I mean, up to a year in here, and then that's good for like storing your genetics, storing your backups for your tissue culture plants. And this is going so fast. This is going really, really fast. Oh man, I haven't done any tissue culture in a little bit before. Last time I did tissue culture, what that was before I moved here to Washington, which has been 2 weeks since I moved, and I probably stopped. I start packing before Christmas, so it has been a while. All right, 1/3 of the way done, and that took me about 10 minutes. So it's probably going to take me like 30 minutes to do all of these plans. So you can see I can do about about 100 test tubes an hour. Let's see my test tube, right? Or my test tubes, a rack. I can take all of this, place it in the fridge. What temperature the fridge? So use keep it on like the higher end. So if a normal fridge is around 4 Celsius, maybe use make it a little less cold. So maybe around under 10 Celsius, so around under 50 degrees Fahrenheit. And that will really, the plants will still be alive, but they're going to slow down. The growth is going to really slow down. So that's what you want, because you want to keep this for a longer period of time. You don't want this to multiply. What you're doing here is just to store the plants. So let me move this out of the way and let me grab the next one. All right. So you see now the rack that I is empty. I'm setting it to the side. I want to work on next. Spray with alcohol. Spray in my hands. And let's get started again. See, that's one of the reasons whenever I was trimming the plants, I always set up to like the this part of the leaves on my right side. So whenever I grab them with my forceps, they are on the right way on. So it's a lot easier for me, a lot faster. So you see, I just grab one, it's on the correct right, or the correct way. So I just go in and then I just place it in the media. Close the lid and go for the next one. So for how long they will be good? Like I say, I mean, depend on the plant, but if you keep them on the fridge, I'll say in general for around a year. And these are mostly backups. So in a year, I will probably take my plants I have in the tubes, place those in like a fresh medium, and then make new ones in test tubes, and then place those in the fridge. Now, these plants in particular that I'm working with, these are not for me for keep it on the storage. I actually have to I will growing these plants. These are for the future class. What is your technique for plant cell culture? Or you don't or I well I have done cell culture, but it's been a long time. I have done that with soybeans. So I mean, it depends on what plant you're working with. So that was back when I was working at the university. Do I use ethanol or alcohol for a wash after bleach and instead of water? No, actually, if I use alcohol, I use the alcohol before I do bleach, and then I do alcohol about 30 seconds to a minute, then bleach, and then I would do the sterile water. But I don't always use alcohol. It depends what plants I'm working with. If I see that the plants are too dirty, like too much soil, or if I'm working, if they have too much soil, then I'll probably clean them. I wash them first in like in the sink with just running tap water and soap, and then I will do a quick alcohol wash, and then I will do the bleach. And then the bleach, I mean, the timing is like, I cannot give you the timing. I can say 10 to 15 minutes, but depends on which plant. I think I'm running out of plants here. I don't think I have enough plants for all the test tubes. That's a shame. That's okay. I can always take more. I'll say the media and then I will do more in the future. Yes, yes, you want to wash the alcohol. You don't want to mix alcohol with bleach. Yeah, definitely. Yes. So yeah, well, you do water to wash the plants first. Then you do alcohol for about 30 seconds to a minute. Then you do water. It doesn't have to be sterile water just yet. Just water, maybe the still water. I mean, if you do sterile water, I mean, that's even better, but it doesn't have to be sterile. Then you do the bleach, and then you do the sterile water. You rinse like 3 times for like 5 minutes each. Yeah, so this is a quick reminder, guys. So we have the upcoming uh, tissue culture masterclass this weekend. So you are interested, make sure to go to plantcelltechnology.com to the website, and you can register for the masterclass there. My class is for houseplants, and then the one of February, that's for hemp and cannabis with Bill Graham. So at the moment, I'm actually growing or moving the plants for him for his class in February. You want us to go live in the future? Let us know. Make sure to leave a like on the stream. We may also stream on Instagram as well, or maybe even TikTok. All right, so this second tray took another 10 minutes. There we go. Boom, second tray. Live video, yes, live video will be amazing. Yes, not only for you, but also for me because I don't have to edit as much. So also another thing about the live streams. I mean, I can do a lot more Q&A, which uh, for me it's easy for to answer questions talking than writing. So if I get an email or a question or a comment, it's a lot more difficult to explain like when I'm writing. So it's a lot easier for me to just to talk with you guys while I'm doing that. Yeah, that'd be nice. Well, actually, I have my other phone as well. Well, I am a zombie today, but yeah, yeah, that's right. I can send my other phone. Right here.
Rich was saying consider it just more effective for like classroom wise,
Yeah, yeah.
while YouTube is good for
Set up. Yeah, more like in a schedule. Go live more often. Get the three cheap phones and do them all. Yeah, I have my second phone, and next time we also do Instagram as well.
tell them about how you work. That the tubes are available online, by the way.
Oh yeah. So again, I mean the test tubes. You can go check plantcelltechnology.com. Uh, you can get this. Use code FP10 for a 10% discount. I mean, as you can see, they are great for doing this kind of work. I mean, it goes so fast. Isn't burner necessary? No, I use a glad bead sterilizer. So what you see right here is a glad bead sterilizer. It's sterilizing my tools. This right now is set to 290 degrees Celsius. So I just need to leave my forceps in the bead sterilizer for about 10 seconds, and my forceps are not sterile.
Oh, like endogenous contamination. That's a good.
Removing endogenous contamination. So yeah, that's oh, we need a video on removing endogenous contamination.
Yeah.
Yeah, ppm. That's what you need. But there are some techniques you can do for reducing endogenous contamination. So that's something that I teach in the class. So if you're interested to sign up for the class, but yeah, I mean it depends on what plans you're working with as well. So well, the plans I teach how to work with that, like monsteras, philodendrons. Oh, I'm cooling down the forceps. Yes, yes. So ideally, I will have two long ones, but the other ones that I have are a little short for test tubes. So and but it's okay. I mean, I have a bunch of water here on my tray, so this is what I do. So you see, I take them out, I cool the tips before I touch the plant. So yeah, you don't want to burn your plants. That definitely will affect the growth. Yeah, yeah, you hear a piece each time you do play the forces in the water. I mean, that's when you can tell that it is hot. They are hot, yeah. All right, one more. I didn't cut enough plants to do all of it. 63, but I got to I don't even know how many, like 58 or so.
That's a good question. Does one plant, does one plant TC become, does one shoot tip become one plant? That's the whole multiplication.
Oh, where is that question? Oh, all right. Let's see. So I'm done. Um, have a few test tubes left, but I am done with that. Okay, there we go. So much quieter now. I don't have to shout. Oh, let's see. If you put a shoot tip to tissue culture, can you get multiple shoots or and get many plants, or those one shoot tip equals one plant? So yeah, one shoot tip is actually it will be equals to one plant at the beginning. Obviously, um, when after it grows, then it will start multiplying. But at the beginning, yes, it's one shoot tip is one plant. But then it grows and then hopefully multiplies, and then you can just cut them and then you go from there. Um, are you dipping your explants to water or alcohol before inoculating? No. So I don't know if you were watching at the beginning, but basically, so I started by let me see. So I started with bleach. I had my bleach. I did my bleach sterilization at the beginning, and then I rinse with sterile water 3 times. And then at the end, I have my sterile water, and then I just dump it on my tray. So all of this, my tray right here is has a use on water, but this water is sterile water, and I do that because I have a lot of explants. I have like close to 60. So I have my explants there in water, sitting in water, so that way they can they um they they don't dry out. And then I was just with my forceps and with the scissors, I was just cutting the very tip where the bleach kill the explant to death. So I was just removing that part, but I was just letting sit in the sterile water. Um, so yeah, that's what I was doing. Um, you can dry them, yeah, but I mean they use sterile water. So yeah, thanks for the explanation. Okay, thanks. All right, guys. So been 53 minutes since we started streaming. I mean, see, in an hour, I mean, I pretty much sterilized close to 60 plants going from you didn't saw the whole sterilization process. You decide to go live like last moment, like, well, I'm about to move plants to test you. So it's like, why don't we go live? So cool. But uh, takes you about an hour and a half to 2 hours to do the whole thing. Well, I would say about 2 hours because I took me some time to actually take the explants and cut them all them out, and then the sterilization process took about just about 30 minutes, and then this took about another 30 minutes. So you see, this actually doesn't take that long, and hopefully I will get a ton more plants from this. Thanks for the explanation. Follow-up questions. If you're using a node cutting, can you get multiple shoots? At the beginning, obviously, one node, one shoot. So that's something that you see a lot with monsteras, where a lot of people ask me, well, what is the point of taking no explants from or like do tissue culture on monsteras, because you start with one node and then you have one plant, and yes, you're going to have one plant for a while. It's going to take several weeks to a few months for the plant to start multiplying. But the point is to get the plant established in tissue culture. You get it established in tissue culture, and then in just basic media, you can use media with no plant regulators. You can start with plant regulators, but it's not necessary. But then you move the plant after is you see that is growing healthy and there is no contaminants, and then you move them to and you move them to media with a lot more cytokinins, so they have multiplication. So um, it can be the gel or it can be like liquid media. So I do liquid media with the biocouplers. I found like with the biocouplers they do very well. Some of the plants like the monsteras. I have a monstera tycon that was growing in agar in gel, and it was growing. It was pretty healthy. The problem it was it was not really multiplying. The multiplication rate was really low. But as soon as I place those in the biocouplers, oh well, actually, hold on, give me 1 second, because I use you pretty much just see in the hood. Let me. Oh, hold on. Hmm. Oh, okay. I think that works. All right. So, um, well, yeah. So, what I was. But yes, it takes the monstera takes some time, but then when I move them to biocoppers, and I start to multiplying a lot faster. Um, Condiment cups you use in a video. Where were those from? So I cannot find them anymore actually. Those cups. But now we have a more. We are now having our own. So like I see, these are brand new. These are test tubes from. Now we have this on the website, like this, and we also have like the petri dishes and deli containers, and well, like the square magenta boxes. So we have all of these available on the website. But yeah, you use like condiment cups. Make sure they're both the lid and the cup are made of polypropylene, because a lot of the times the cup is made of propylene, but the lid is made out of other material, most likely PET, and then it gets whenever you sterilize it, it just melts. Mm. do I supplement any antibacterial or antifungal agent into your meal? Yes, I use ppm, plant preservative mixture. So that's basically your flagship product here at Plant Cell Technology. That's the product that basically at one point I would say that it was bigger than the company, that people would know what a ppm was, but it wasn't didn't really know that about plant cell technology until I think now that we have the classes, we are known for the classes and the YouTube and the content, and we also have like media and some other products as well, and not just ppm, but ppm still is our flagship product, though, that what we sell the most here. Thanks for the amazing content. Well, thank you so much. Thank you so much. Uh, Protocols on cannabis. I mean, I use moving some plans right now, but we had a class with Bill. That's in February. So if anybody interested on that, you can check that in the website as well. You can use my referral code to get a 10% discount. Um, After inoculation of fresh explant of using cotyledons, is, why is it recommended to incubate in dark conditions for 48 hours for callus induction? Hmm. So, at least with soybeans, which is the plant that I was working with back in the day, you place it in the dark because if you don't want it to grow shoots, at least that will happen with a lot with soybeans or with um, rice. So we would do the rice callus. We'll grow the callus in the dark, because if we put it on the in the light, then we'll start making sending shoots, which we didn't want. We want the callus because we needed enough callus to use agrobacterium to transform the plants, because we were doing genetic engineering plants, so like your GMOs. Um, so that's why we were use. We were keeping in the dark. Uh, but that the protocols is there are so many different protocols for different plants. So all right, I think it's been an hour, so I'm done with all my transference for today. So I think I'm going to stop the stream. Like I said, it was just a test stream, but you guys enjoy it. Please leave a like. I think if I do this, it will actually give you a like, pop up a like. I don't know. Um, but yeah, please leave a like, and then let us know you want us to do more. I mean, I really enjoy this. It's something like I said last minute. It's like, oh well, let's do a live stream. So and there we go. So you just do a live stream. It's pretty quick, pretty easy. I have a really nice setup. I made you like a quick short video on like my setup live stream. Yeah, there we go. Okay, I see now that it pops up like a thumbs up. So there we go. Excellent. So all right. Well, thank you so much. Hope you have enjoyed it, and then I will see you next time. Goodbye. Well, actually, let me see. How do I end the stream now? All right. So let me take a look here. First time streaming using OBS on YouTube. I don't even know how to end the stream. Let me click the here. All right. All right. So I found the button. All right.